Hello, everyone. Thanks to the organizers. Actually, I have got a bad cough these days, so maybe I have to go through the slides briefly, and we may discuss it later. Sorry for that. Actually, some of work in this study we had then attributed to my supervisor, Professor Daoqiang Zhang at Nanjing University of Aeronautics and Astronautics, and my visiting supervisor, Dr. Li Shen, at Indiana University. The title is Identification of Associations between Genotypes and Longitudinal Phenotypes, and we named our proposed method as the Temporally-constrained group sparse canonical correlation analysis. And here is the outline of my presentation. First is background and motivation. The second is our proposed method, TGSCCA. Finally, we draw the experimental results on both simulation and ADNI dataset. There is a special report about Alzheimer's facts and figures from Alzheimer's Association. As the population of United States ages, deaths from major causes such as breast cancer, prostate cancer, heart disease, stroke, HIV have decreased significantly, but official records indicate that Alzheimer's disease increased 71%. So first, what is AD? Alzheimer's disease is the most common form of dementia, decline in memory, language, problem solving, and other cognitive skills. There's no cure for the disease, which worsens as it progresses and eventually leads to death. So research on AD is very important for possible delay and early treatment. Alzheimer's Disease Neuroimaging Initiative is a global research effort that supports the investigation and development of the treatments. So our research has also focused on exploring more sensitive methods for early diagnosis of AD and finding effective biomarkers to track its progress. As known from biomedicine, Alzheimer's disease is also considered as a genetic relevance disease. Intermediate phenotypes such as gene expression, brain imaging, and clinical records are associated with the genotype single nucleotide polymorphism. To investigate the mechanism of the disease, some researchers have developed a new research field, imaging genetics. So what is imaging genetics? Imaging genetics are interested in how genes influence psychopathology and using functional neuroimaging to investigate the genes that are expressed in brain. So the goal is to investigate the role of the genetic variations in disordered function using neuroimaging and biomarkers as the phenotype. So motivation. Most existing approaches are used to detect effective associations between static genotype and static phenotype at one time point, such as sparse canonical correlation analysis. But the problem is that the genotype could be related to the static phenotype that is knowing the temporal dynamics. Actually, we also have the time 1, time 2, time 3, and time 4 for the longitudinal phenotypes. So a powerful model would be to discover the longitudinal imaging genetic associations and capture the consecutive changes in brain from adjacent time points. Assume that we have known candidate loci and longitudinal phenotypes with regions of interest. How can we discover the associations between them? To address the above issue, we extended sparse canonical correlation analysis to this formulation. Here is the object function. The first term is maximal correlation, and L1 norm is sparsity on SNPs, and L21 norm is group sparsity on regions of interest, and the last term is filter penalty from adjacent time points. Of course, the constraint is the scaling from the original formulation. The object function is convex with respect to U when V is fixed and vice versa. So the iteration procedures mainly contain two steps as follows. Here is the algorithm about this our proposed method. For more details about it, we can discuss this later. And we also set up a simulation study to test the performance of our proposed method. Firstly, we generated vector U and V with some non-zero elements, and then each element in U and V was sampled from a uniform distribution, and features were simulated from Gaussian distribution, where a latent variable H from normal distribution. And finally, we set up a simulation data 1 and simulation data 2 with different noises. This figure shows the average correlation coefficients on five-fold test data using different methods on simulation data 1 and simulation data 2. Here, GSCC means the group sparse CCA without filter penalty. And we can see that GSCC and TGSCCA outperform SCC on both simulations. But TGSCCA can get the best performances on simulation 2 when there is more noises in this data set. This figure shows the feature selection panels on simulation 1 and simulation 2. This one is the ground truth on U, and this ground truth on V, and this also. And others are estimated from different methods. We can see that our proposed TGSCC can capture the smooth patterns, which is more consistent with the ground truth. In ADNI experimental data, we use the longitudinal imaging phenotype MRI and SNPs around APOE. Diagnosis label include PMCI, SMCI, NC. Here, MCI is a pre-stage of AD. This figure shows the characteristics of the subjects. NC means normal control, PMCI means progress MCI, and SMCI means stable MCI. They yield all 114 subjects. And this figure shows the average correlation coefficients on five-fold test data using different methods on ADNI. We can see that our proposed TGSCC also can also get the best performance, which is accordance with the previous results in the simulation study. So this figure shows the feature selection results on both SNPs and the brain features. So we can see that our proposed method can select more smooth features, which is accordance with a previous study in literature. And this further indicates that our proposed method can capture the consecutive changes in brain from different time points. Okay, to summarize, this study developed TGSCC to develop the risk genetic factors and their correlated longitudinal phenotype markers for progressive disease. And also, this framework can be extended and applied to other interesting fields, such as brain and gene expression analysis. Acknowledgement, our teams at NUA and IU. Also, travel fund was supported by ISCB. Okay, thank you. Yeah.
And does that affect what happens with the process by which you do that?
Yes, actually, we use this U for our one norm U for the feature selection on the SNPs. Yeah.
What about in terms of the ADNI data, extracting features from the images, the brain images?
Yeah.
How does that happen?
You mean that the associations between? Yeah. Actually, we formulate the sparse canonical correlation analysis on this. So how to say on this mechanism of the disease. Actually, we just try the model for feature selection. Yeah.
Well, thank you very much indeed.
Yeah.
Oh, one more question, please. We have time.
Why did you use only 85 SNPs? Because you have the sparsity term, so you should deal with that, right, with the SNP selection.
Yeah.
You use a very small number of SNPs. What was the reasoning for picking only those?
Yes, actually, this is our primary experiment, and we use the ApoE as a SNP. Maybe in the future, we will extend it, the last set of the genetics and the brain regions.
Because SNP selection, I think, SNP selection is also a problem which is relevant in this context, right?
Yes, it all depends my algorithm. Actually, the complexity of the algorithm is related to the features. So I think it's promising to extend our model to large problem.
Because of matrices or?
So sorry?
Oh, okay.
Yeah.
Never mind. So you're saying you're going to extend it to deal with more SNPs?
Yeah. I think it's a general model to apply to a large data set. Okay, thank you.